steroids are further classified into adrenal cortical hormones and sex hormones. If vitamin A is present in lesser amount, it can cause night blindness, which is called as xerophthalmia. In DNA, it is thymine which is present. In RNA, it is uracil which is present. So you have adenine, guanine, uracil and cytosine which is present in RNA. It has a double helical structure in DNA and it has a single stranded helical structure in case of RNA. Hello everyone, this is Ambli Unnikrishnan from the Department of Chemistry. So today I'm back with session 5, that's the last session of Biomolecules chapter. So in the last session, we discussed about proteins and the structure of protein, that is primary, secondary, tertiary and quaternary structure of protein. And then we moved on to the denaturation of proteins as well as we discussed about enzymes. So in today's session, we are going to discuss about hormones, vitamins and nucleic acids. So nucleic acids are your DNA and RNA. So you have a basic idea about it in your 10th standard itself. In biology, you have studied, right? Okay, so moving on to hormones. So hormones are the proteins secreted by ductless glands of the body. Okay, so you might be familiar with hormone imbalances taking place, right? So actually they are also proteins which are secreted by specific glands of your body. They enter the blood and initiate the required physiological activity. Thus, they are called as biochemical messengers. Okay, so each of these hormones are having a specific function to perform and each of these hormones are secreted from a specific area of the body also, that is from the specific glands. So each of them have a specific physiological activity to perform. So you can see pituitary gland, pineal gland, thyroid gland. So thyroid gland secretes the thyroid hormone which is important for your body as well. So if the amount of the thyroid present is less or more, it can cause hypothyroidism or hyperthyroidism, which is having a major effect on your body, right? And you have thymus and adrenal gland, pancreas, ovary, testis. Testis is present in male and ovary is present in females. All of these are important glands which has the function to secrete certain hormones from each of these glands which are important for our body, right? So let's see how the hormones are being classified into. It is classified into steroids. So steroids are further classified into adrenal cortical hormones and sex hormones. Right, adrenal cortical hormones and sex hormones. And sex hormones are further classified into female and male sex hormones. So male sex hormones, you have androgens and testosterone. Testosterone you are familiar with, and in female sex hormones you have estrogen and progesterone, right? Estrogen and progesterone, right? And you have on the other side you have peptide hormones which is insulin, glycagon and amino acid derivatives you are having, and miscellaneous, that is prostaglandins and cytokinins. Okay, so this is how the classification of hormones is done. So under steroids you are having adrenal cortical hormones and sex hormones. Sex hormones further classified into female and male. And male you have testosterone, in females you have estrogen and progesterone. I hope it is clear. Alright, now some important types of hormones and their functions, let us see. So polypeptide type, you have insulin and glucagon. So polypeptide, while discussing about proteins also we were discussing globular protein. Insulin is a globular protein, right? So what is the function of this insulin? It helps in maintaining the blood sugar level. So those people who are suffering from diabetes, if their sugar level is very high, they usually take insulin shots, right? So why insulin shot is taken? It will help in regulating the blood sugar level in our blood, right? So it is very important to have the particular amount of insulin in our body so that it will maintain the blood sugar level. So if sugar or diabetes is more, that is the blood sugar level is more, insulin will help in maintaining that level. So when in the body the insulin level is less, that is when the patient or the person is getting diabetes. So when it is the level of the diabetes is more, they take the insulin shots which will regulate the blood sugar level. Next is amino acid derivatives hormones. You have epinephrine which brings out the response to stimuli. How we respond to various stimuli, that is based on epinephrine. And then you have steroids, in that which estrogens and androgens, which is development of secondary sexual characters in the human beings. So estrogen helps in the development of secondary sex characters in females and androgen in that of males. So these are few important hormones. Moving on to vitamins. Vitamins are organic compounds required in the diet. Okay, they are required in the diet in small amounts to perform specific biological functions for normal maintenance of optimum growth and health of the organism. So vitamins are very, very necessary for our body. So you have heard about taking vitamins tablets also, right? So they are really important for the functioning of human body. So they are taken through our diet, that is in each of our foods there is a specific vitamin which is present, which is important for your body, right? So it is necessary to consume such fruits or vegetables or green leafy vegetables so that you get the specific vitamins, right? So it is important for the growth and maturity of our body, right? So the classification of vitamins, they are classified into two groups depending upon their solubility in water or fat. Okay, so you have fat soluble vitamins and water soluble vitamins. So these vitamins are soluble in fat and oils but insoluble in water, right? Now water soluble, of course they will be soluble in water. Now they are stored in liver and adipose tissues. Okay, so they are stored in liver and adipose tissues, that is fat storing tissues. The fat soluble vitamins are stored there, and water soluble vitamins must be supplied regularly in diet because they are readily excreted in urine and cannot be stored in our body except vitamin B12. So fat soluble vitamins have the ability to be stored in our adipose tissues or liver. So it is not much of a problem. But water soluble vitamins have a problem because we have to take it more through our diet, because as it is water soluble vitamin, what happens when the urine is excreted, through that the vitamin also will be lost. So as to reduce the loss of this vitamins, it has to be consumed more or regularly through our diet. They are called as water soluble vitamins. Example, vitamin A, D, E, K, all of these are example of fat soluble vitamins. And you have vitamin C and B group vitamins. You have B1, B2, B6, B12, all of this which comes under B group. All of them are water soluble, which are important to consume through diet regularly. Now, vitamin sources and their deficiency diseases, let's see. So vitamin A, from where you can obtain vitamin A? They can be obtained from fish liver oil, carrots, butter, milk, etc. Okay, carrots are a very good source of vitamin A, right? So now moving on to the deficiency diseases. Deficiency means what? If it is not present in our body in the required levels, you will cause or you will have a deficiency of that vitamin, which can cause particular diseases. So if vitamin A is present in lesser amount, it can cause night blindness, which is called as xerophthalmia. Okay, night blindness, that means in the night you won't be able to see, that is vision will be not much clear, right? Now vitamin C, it is present in citrus fruits, amla, green leafy vegetables, that is a source, and the deficiency disease is called as scurvy. So these are important to remember, which is the vitamin, which is the source from which you can obtain this vitamin, and what is the disease that will be present if it is present in lesser amounts, that is if there is a deficiency. Moving on to vitamin D, which is present in fish, egg yolk, exposure to sunlight. So vitamin D is very important, from where you can obtain it from the major sources from sunlight. So it is usually recommended in the early hours, that is in the daylight, in the early hours, in the morning, not during the afternoon time. Morning time it is recommended to go outside and catch hold of the sunlight, because it helps you in getting the vitamin D, right? Now the deficiency diseases, rickets and osteomalacia. Now you have vitamin E, which is obtained from sunflower oil, wheat gram oil, etc. And the deficiency diseases, muscular weakness and increased fragility of RBCs. RBCs is red blood cells. Now moving on to vitamin K, which is obtained from green leafy vegetables, and the deficiency causes increased blood clotting time. Blood clotting means what? Usually if you have an injury, what happens? If your hand get cut somewhere, the blood will start flowing. After some time, what will happen? It will stop flowing out, right? The blood will stop flowing out because our body itself will act upon it and clot this blood. Okay, so if you are having a deficiency of vitamin K, what happens? It will increase this blood clotting time. That means more amount of blood will be lost from your body. The body will take a more amount of time to clot the blood. Clear? Now, next one is vitamin B1, yeast, milk, cereals, green vegetables. From all of these you can obtain. And deficiency causes beriberi disease. Now, moving on to vitamin B12 obtained from milk, egg white, liver, etc. And the deficiency causes scoliosis, digestive disorders and burning sensation of the skin. Now, vitamin B6, it's present in yeast, milk, egg yolk, cereals, grams. Those are the sources. And it causes convulsions if you are having a deficiency of. And you have vitamin B12, which we have studied in coordination chemistry chapter also, right? Vitamin B12. So the sources from which you can obtain vitamin B12 is fish, meat, egg, curd, etc. And the deficiency causes pernicious anemia. So that is, you will get anemia if there is a deficiency of vitamin B12. Anemia means what? There is less count of RBCs in our body. Right. Moving on to the last part, that is nucleic acids. Right. So nucleic acids are long chain polymers of nucleotides. So they are called as polynucleotides. Nucleic acids are also called as polynucleotides because many nucleotides units will be bound together by long chain polymers and form nucleic acids, which is called as polynucleotides. Okay. So what are nucleotides and what are nucleosides? All of this we are going to study now. So there are two types of nucleic acids. One is deoxyribonucleic acid, which is your DNA, right? DNA. And you have ribonucleic acid, which is RNA. So regarding this, you have studied in 10th standard biology itself in detail. So we are going to study the chemistry part of it. They are responsible for protein synthesis and a transfer of genetic characteristics to the offspring. So DNA will help in transferring the genetic characters to the offsprings. That is why we look similar to our parents, right? So that is about DNA, and RNA helps in the transfer of protein or the synthesis of protein and the transfer of these proteins. That is done by RNA, and the genetic details or genetic characteristics is passed on to the offsprings through DNA. Now let's study about the chemical composition of the nucleic acids in general, that is both in DNA and RNA. They are made up of pentose sugar. So first thing you have is a pentose sugar. So this pentose sugar can be two, that is alpha D deoxyribose in DNA and alpha D ribose in RNA. So it is alpha deoxyribose in DNA and alpha D ribose in RNA. So that is the pentose sugar. Pentose means 5, right? So pentose sugar. So it can be either deoxyribose or ribose in DNA and RNA. And the next part is you have a phosphoric acid group. Okay, you have a phosphoric acid group and you have one more part, that is a nitrogen containing heterocyclic compound, which is a nitrogenous base. Okay, that is a base which contains nitrogen. It is a heterocyclic compound. That means it is not just containing carbon, a nitrogen is also present. That is why it is called as a nitrogenous base. So I hope you understood. The basic structure of nucleic acid contains 3 major parts. One is the pentose sugar. In DNA it is deoxyribose. In RNA it is ribose. Then you have a phosphoric acid group, right? And then you have a heterocyclic nitrogen containing base, which is called as a nitrogenous base. Okay, so in DNA the bases present are, so this nitrogenous base will vary in case of DNA and RNA. So in DNA the nitrogenous bases are adenine, thymine, guanine and cytosine. Is this adenine, guanine, thymine and cytosine are the 4 nitrogenous bases present in DNA. So now let's say the case of RNA, you have adenine, you have guanine, you have cytosine and you have uracil. So what is the difference here? In DNA you have thymine. In RNA instead of thymine you are having uracil. So other 3 are same, right? You have adenine, guanine, cytosine. You have adenine, guanine, cytosine. So in DNA it is thymine which is present. In RNA it is uracil which is present. So you have adenine, guanine, uracil and cytosine which is present in RNA. So this is very important to remember. The nitrogenous base is present in DNA and RNA. The one which differs is thymine in DNA and uracil in RNA. Clear? Now moving on to the nucleoside and nucleotide. So while we were discussing about the introduction to nucleic acids, We have already discussed that it is a long chain polymer of nucleotides. It is a long chain polymer of nucleotides. So what exactly are those nucleotides? Let's see. Before that, we need to understand about what is nucleoside, which will form the nucleotide. So what is nucleoside is? It is basically a combination of your pentose sugar and base. That's it. The unit formed by the attachment of a base to the first position of the sugar, that is the base plus sugar, that nitrogenous base plus the sugar is called as the nucleoside, right? In nucleosides, the sugar carbons are numbered 1, 2, 3, etc. in order to distinguish from the bases. So in sugar, that is your pentose sugar, the carbons will be numbered as 1, 2, 3, and in nitrogenous bases, it will be numbered as 1 dash, 2 dash, 3 dash, etc. to differentiate the numbering of the carbon. So I hope it is clear. What is nucleoside? That is your base, nitrogenous base plus sugar. Okay, this part is called as nucleoside. Now, how is nucleotide is obtained is, so the nucleoside plus the phosphoric acid group will give you nucleotide. See, nucleoside and the phosphoric acid at the 5' position. Nucleotides are bonded by phosphodiester linkage between 5' and 3' carbon atoms of the pentose sugar. That is all together. That is your base plus the sugar plus the phosphoric acid. All of these three together will form the nucleotide. I hope it is clear. So it is how this nucleotide is formed is by the phosphodiester linkage, which is formed between the 5' and 3' carbon atom of the pentose sugar. So let us see here. This is your nitrogenous base and you have the pentose sugar. So these two are combining together to form what? Nucleoside. Now this nucleoside is going to combine with or form a phosphodiester linkage with what? It is going to form a phosphodiester linkage with the phosphoric acid group, which will give you what? The nucleotide. So you can see this is the phosphate group which is present. Right, phosphate group, and you have the sugar and you have the base. Okay, the base used here is adenine, and you have the pentose sugar and you have the phosphoric group. So all of this three together, what you are getting, this is called as your nucleotide, and this nucleotide will form a polymer chain, long chain polymer to form what? Nucleic acid. Right, now let's see here. This is the phosphate group you have, and this is your sugar, and this is the base. One nucleotide. This whole part is one nucleotide, and you have another nucleotide here. Now what is going to happen? You can see this is the 3' position of the sugar and this is the 5' position of the next sugar. Right. This is one sugar and this is another sugar. This is a 3' of first sugar. This is the 5' of next sugar. So what is going to happen? A phosphodiester linkage. It's through the phosphate group that this bond is formed. That is why it is called as phosphodiester linkage. So it is formed between the 3' and 5' position of the two sugars or the two nucleotides. Clear? So this is how the nucleotides is forming phosphodiester linkages and forming the nucleic acids. Now let's understand the structure of the DNA. Okay, double helical structure of DNA or the double helix structure of DNA. Right, it is composed of two right-handed helical polynucleotide chains coiled spirally in opposite directions around the same central axis. So you can see the structure here. There is a central axis here. So you can see the pink color as well as the white color here. So imagine it as two different strands and there is a center of axis in the middle. So one is coiling in this direction and another one is also coiling. So what is happening? You are getting a double helical structure. So only one is called as a single helical structure. On the other side, you have another strand which is coiling from the other side also. That is why it is called as a double helical structure. So you can see here, one is coiling in this direction, another one is starting from this side and coiling in the other direction. That is why it is called as a double helical structure. Now, these two strands are anti-parallel. That is, their phosphodiester linkage runs in opposite direction. They are not parallel to each other. That is, these two are coiling in opposite directions and these two strands are held together by hydrogen bonds. Okay, so you can see here also these are two strands which is coiling in opposite directions. But how these two strands are present in that same position without getting uncoiled? That is because of extra hydrogen bonds which is present. So this hydrogen bond is formed between adenine and thymine, guanine and cytosine. Only in that form it will be formed. Okay, so between adenine and thymine that is a double bond, and guanine and cytosine in case of DNA. Now we are discussing the structure of DNA, right? But if in the case of RNA, if we have to discuss, it is going to be between adenine and uracil, guanine and cytosine. Clear? Right. So you can see here, this is one strand, this is another strand. Between guanine and cytosine, there is a hydrogen bond. Between adenine and thymine, there is another hydrogen bond. Between thymine and adenine, between adenine and thymine, there is hydrogen bonds which is present. So because of this bonds which is present, what is happening? The strands will remain in that position. That is how this double helical structure is maintained. Right. And only the hydrogen bonds will be formed between adenine and thymine, guanine and cytosine. There is no possibility of adenine and guanine having a hydrogen bond or thymine and cytosine having a hydrogen bond. A and T will combine, G and C will form the hydrogen bond. Now the two strands are complementary to each other because the hydrogen bonds are formed between specific pair of the bases. Adenine forms hydrogen bonds with thymine, whereas cytosine forms hydrogen bonds with guanine. So that is fixed, it won't change. Now, in secondary structure of RNA, helices are present in which there are only single stranded. So, DNA it is double helical structure, RNA is single helical structure. It has only one strand. Sometimes they fold back on themselves to form a double helical structure. So, you can see this is a double structure. Two strands are coiling with each other, that is double helical structure which is in DNA. But in RNA, that only one single strand will be present which will curl upon itself to form a double helical structure sometimes. Clear? Now, RNA molecules, they are of three types and they are having different functions. The first one is messenger RNA which is called as mRNA. Then you have ribosomal RNA which is called rRNA, and transfer RNA which is called as tRNA. So these are the three types of RNAs, that is mRNA, rRNA and tRNA. Clear? Now let's conclude this by the difference between DNA and RNA. So DNA's sugar moiety is 2-deoxyribose. Already we have discussed it is alpha deoxyribose which is the sugar present here. In RNA it is just D-ribose, alpha D-ribose. It is deoxyribose here, ribose here. Right. Now, it contains thymine and cytosines and pyrimidine bases and guanine and adenine as purine bases. So, we have already discussed about the different nitrogenous bases, right? Adenine, guanine, thymine, cytosine and uracil. Now, they are further classified as pyrimidines and purines. So, the pyrimidines are thymine and cytosine in case of DNA, and the purines are guanine and adenine. Okay. In case of RNA, that contains cytosine and uracil. Uracil as the pyrimidine and guanine and adenine as the purine bases. So, you have the purines which are adenine and guanine. These are the purines, and you have pyrimidines which are thymine, cytosine and uracil. So, in case of DNA, it is adenine, guanine which is the purines, and pyrimidines will be thymine and cytosine. And in case of RNA, the purines will be adenine and guanine, and the pyrimidines will be cytosine and uracil, not thymine. I hope it is clear. Now, it has a double helical structure in DNA and it has a single stranded helical structure in case of RNA. Chiefly occurs in nucleus of the cell and mainly occurs in the cytoplasm of the cell, right? Because it is present in cytoplasm of the cell because it helps in the protein synthesis and the transfer of it, right? It has unique property of replication. It has the unique property of replication, that is why it is able to produce or transfer its genetic characters to the offspring. So, it will replicate, right? And it does not replicate. RNA does not replicate, and DNA controls transmission of hereditary effects and it controls the synthesis of proteins. So, it will help in synthesis of protein and the transfer of the proteins, whereas DNA deals with the genetic things, right? Now, moving on to the biological functions of nucleic acid. DNA is the chemical basis of heredity and may be regarded as the reserve of genetic information. For each human being, the DNA is unique, right? And because of this presence of DNA, the information will be transferred to the offsprings, and that is why we all look similar. That is, we look similar to our parents because of this genetic information, right? A DNA molecule is capable of self-duplication during cell division, and identical DNA strands are transferred to the daughter cells. So, DNA has the capability to undergo replication and form daughter cells. Your RNA will synthesize protein in the cells. Actually, the proteins are synthesized by various RNA molecules in the cell, but the message for the synthesis of a particular protein is present in the RNA. So what does that mean? The information or the message regarding the protein will be present in the DNA, but RNA will be the one which is going to synthesize it. So all the information will be present in DNA. The one which is going to produce the protein and transfer the protein will be RNA. I hope it is clear. So by this we come to an end to the chapter Biomolecules. So it is a small chapter which is having certain important topics as well, right? So I hope the concepts that I have covered in this chapter is clear for you. So in the next session, let's meet with a new chapter. So that's all for today. Thank you.